So what Andreas and I are going to do over the next 30 minutes or so is just give an update as to where we are with medical treatment of Dravet syndrome, and then go a little over studies that are planned, not least the natural history study, and then moving on to genetic therapies. I have to say it is quite exciting times as professionals that we seem to be in this position, and I hope that you find the talk presentation informative and useful. But then, of course, we're happy to answer questions at the end. So where are we with treatment? And I probably don't need to explain to this group in detail. But first of all, just to summarise as to what we're talking about, and the fact, of course, Dravet syndrome, as I said, the experts are online, really. But an early onset developmental and epileptic encephalopathy that comes, presents with the first seizures in general in the first year of life, usually long seizures, one-sided seizures, seizures associated with fever, with the later appearance of different seizure types. And we know that developmental delay becomes progressively more apparent. And we know that more than 80% are now associated with a mutation in the sodium channel gene SCM1A. This is not just a childhood disease. We know that there are adults, and we know that seizures are an ongoing problem into adulthood. The issue that less adults we appear to be aware of less adults is because of the fact that most of the adults we know have grown up with a diagnosis, whereas actually there are many adults out there probably who have the diagnosis, but they haven't been diagnosed yet. And as from this European study of in excess of 500 individuals, you can see that looking at the responses, which were from patients and carers right across the age range, there were only 6% who had been seizure-free for the previous 3 months. And this just gives, as I said, I don't probably need to say this to many of those on this call, but this gives the average, the general developmental trajectory, the IQ as with age of individual patients with Dravet syndrome. The IQ dropping, not because they're losing skills necessarily, but because they're not making the same progress relative to normal peers. So over time, with defining this syndrome, we've begun to know what medications may be most useful. Obviously, we don't seem to have an optimal treatment as yet, but what medications appear to be most useful in managing the seizures. And where we have valproate and clobazam with stiripentol, topiramate, levetiracetam, ketogenic diet, and bromide. And we've also learnt what medications are contraindicated that we don't want to use in this syndrome, not least because of their mechanism of action and the fact that they're what we call sodium channel blockers, carbamazepine, phenytoin, lamotrigine, and these may aggravate seizures. But, we have to remember that actually the trial of stiripentol in a randomized controlled trial, in addition to clobazam and valproate against placebo, was one of the first randomized controlled trials in a small population of a complex epilepsy syndrome, made possible because of the well-defined clinical features. And this demonstrated without doubt that addition of stiripentol led to significant benefit in seizures over and above an addition of placebo to a non-active treatment of valproate and clobazam. But this was now 20 years ago, so we've come a long way in addition since then. This aside, we know now that stiripentol doesn't seem to lose its effect. It retains effect over time and is well tolerated. There is some data to looking at whether you can predict who may respond better to stiripentol than others. And remembering that Dravet syndrome is actually a clinical diagnosis from the pattern of seizures, not a genetic diagnosis. Certainly there's a suggestion that the efficacy or the effectiveness of the stiripentol is greater in those with the SCM1A mutation. And as we gain more experience of use in adults, there seems to be, adults don't seem to respond as well, but and there is some evidence that maybe, although looking at group data, that there may be a similar tolerability profile, it can be more difficult to fine tune in the adults. Data with regard to other medications that have been found to be useful really isn't from randomized controlled trials, it's from open label, which means they've just been added onto medication, and then we've the effectiveness has been reviewed looking at a cohort of individuals, and there's data on topiramate, levetiracetam, rufinamide, and even more recently perampanel, suggesting benefit where other drugs have failed. Bromide is a relatively old anti-epileptic drug, the first anti-epileptic drug that was utilized from the early 1900s. But actually, even though it's not a common anti-epileptic drug that's used, certainly it's one that's been found to be useful, particularly where other drugs have failed, in treatment of seizures associated with Dravet syndrome. This from a German study, but we also utilize it in the UK. And the ketogenic diet in this study shown to be as effective as, almost as effective as valproate, clobazam and stiripentol, and again something that can be utilized perhaps when first-line medication has failed, if the individual is able to tolerate the diet. We learned relatively early that there are treatments we need to avoid, as I've already mentioned. This was the first publication from Italy as long ago as 1998, where they spotted that actually utilizing lamotrigine led to aggravation of seizures, whether it be convulsive seizures or myoclonic seizures. And this has since been replicated in other studies, not least by the Glasgow group. And there is some suggestion now that actually utilization of contraindicated medication in the first 5 years of life may have an impact on the natural history. And this just looking at a whole series of children and their developmental trajectories with Dravet syndrome, and those who had not been on carbamazepine doing better than those who had. It's never too late. And in diagnosing a series of adults who'd never been diagnosed with the syndrome, the oldest of whom was 60, 7 had changes following diagnosis, 3 had meaningful follow-up, 2 an improvement in cognition, and 1 spontaneous language for the first time, and that was the 60-year-old. So it's never too late for the diagnosis and for the changes to be made. That said, we also have to be cautious, because as this publication suggested, there are some individuals who seem to respond or be maintained on lamotrigine, and these 3 individuals had an exacerbation of seizures on withdrawal of lamotrigine, albeit older individuals. And there are of course, all the practical aspects that we have to think about: the prompt treatment of fever, being aware of triggers, and also prompt treatment of prolonged seizures with individualized rescue care plans. Although I'm not going to go into algorithms with regard to treatment of long seizures today. What about the newer agents? And there's been much excitement, of course, about cannabinoids, whereas the data is really for the use of cannabidiol, pure cannabidiol, in the treatment and in these studies and treatment of epilepsies associated with Dravet syndrome. And this, the first study comparing 1 dose of cannabidiol against placebo, showing a significant reduction in seizures at the end of the treatment and the maintenance periods. And this, the second study published last year, where they compared 2 different doses, a lower and a higher dose of cannabidiol against placebo, and found actually no additional benefit with the higher dose of cannabidiol, although the prevalence of side effects was actually seen to be, of course, more in the higher dose. It's relatively well tolerated, drowsiness, diarrhea, and decreased appetite being the most common side effects with cannabidiol, but actually those that having adverse events leading to withdrawal of the medication are relatively small. And we now also know with the open label extensions that actually any benefit seen appears to be sustained. There's been a lot of discussion about how cannabidiol may act, and one of the early proposals was whether it was due to the interaction with clobazam. When added to clobazam, the metabolite of clobazam from the liver increases, norclobazam. Cannabidiol impairs the enzyme that breaks down the clobazam, and we get a rise in levels. And that can lead to some of the side effects, but other data now suggests that if they're already on stiripentol, the increase in the clobazam metabolite levels is not seen, and therefore suggests that actually stiripentol saturates the system in the liver and does not lead to the same. And in fact, much of the data now suggests cannabidiol has its own anti-epileptic effect. Now, of course, the other new medication on the block is fenfluramine, and now there's been a European license for the utilization of fenfluramine in Dravet syndrome, and it's going through the NICE HTA process currently. The data from 2 randomized controlled trials is extremely promising in the fact that on the left, we have the study when children with Dravet syndrome were not on stiripentol, and 2 different doses of fenfluramine were compared against placebo. And on the right, those in children who were on stiripentol, they had a slightly lower dose of fenfluramine, again compared to placebo, and a significant benefit noted and in the fenfluramine groups against placebo, and the difference between the placebo group and the fenfluramine group being much bigger than we've seen in previous anti-epileptic drug trials. Of course, we have to be aware of any treatment emergent adverse events, and this decreased appetite was seen but was not common. And also a decreased weight, although the relative prevalence of that was not dose related. Now, one of the issues and why there's been a lot of monitoring of this medication is when it was utilized in the 1960s as an anti-obesity drug, that it was withdrawn because of cardiac and side effects. And so there's been intense cardiac monitoring, heart monitoring through the trials. And to date, being aware much lower doses of fenfluramine are being used here than were used originally, no cardiac or pulmonary problems have been noted to date, which is extremely reassuring, but there will need to continue to be monitoring. There's also the discussion about, is it only the seizures that are improved? And certainly in the trials, an inventory of executive function was undertaken, and improvements in subscores noted in fenfluramine treated patients, not seen in placebo patients. But of course, the time duration of the trial was extremely small. And some other data that we've extracted has been with regard to SUDEP, in the fact that actually looking at all the data across the trials, the rate of SUDEP pre-use of fenfluramine was the same as seen in other cohort studies, whereas it seemed to be dramatically lower in the fenfluramine treated groups, possibly related to the reduction in convulsive seizures, but there's some animal model data that suggests there may be other mechanisms as well. So then we come to our algorithm of what we may use for treatment of Dravet syndrome, particularly the seizures. And yes, the general principle is still that sodium valproate would be used first, and that stiripentol be added with or without clobazam. And at the present time, it may be considered that cannabidiol would be the next in line treatment. We await in England for the NICE HTA decision, but would presume if fenfluramine is approved, then it would be quite high up in the second line management. Being aware, of course, also that other medications are there to utilize, and including the ketogenic diet, should first and second line treatment fail. And with that, I'll hand over to Andreas, who's going to talk about the studies.
Thank you very much, Helen. That's thank you for that really nice overview. So what I would like to start really with talk a bit about the SCN1A Horizon study. So this is a natural history study of SCN1A related epilepsies in the United Kingdom. And so if you move to the next slide, Helen, thank you. Yeah, so I think we now know that Dravet syndrome is really what we call a developmental and epileptic encephalopathy. And what we mean by that is that Dravet syndrome not only causes seizures, but we know that individuals with Dravet syndrome also have difficulties with movement. It might be unsteady. There's difficulties with learning. There's difficulties with other behaviors, autistic behaviors as well. So I think we understand that now Dravet syndrome is really a whole group of different symptoms and presentations, and what we're really interested in to really how we can actually monitor individuals with Dravet syndrome, children as well as adults, in all these different aspects, because we have good data on seizures, but I don't think we have enough data and good enough data on all the other aspects of Dravet syndrome. Next slide, please. So Dravet syndrome really is one of the most common childhood genetic epilepsies, or monogenic epilepsies, where there's one particular gene defect causes epilepsy. And as Helen earlier explained, we've now got a number of drugs that are effective at reducing the seizure burden, but we really don't understand their impact on cognition. And as Helen explained from the latest fluorine trials, that there was some evidence that it might have an effect on executive function. However, the follow-up time was very small, so we don't really have good enough data and to know how these medications might actually affect the development. And I think without understanding what we call the natural history step, by the natural history, we really mean to understand how an individual with Dravet syndrome evolves over time. And by studying this, we really get a much better idea of what all the characteristics really are. And without that, we can't really, without those data, we can't really measure any treatment success just beyond seizure control. And that's why we really need to characterize the disease in much more detail. Next slide, please. So we've, I think now over the last year and a half, almost 2 years, we've been really getting together with lots of child and adult neurologists across the United Kingdom and 27 different centers to really come together and plan this natural history study. And we think that if we involve lots of children from all over the United Kingdom, this will allow and adults, that will allow all these individuals to actually have some access to standardized assessment. And by that, we think if any of individual with Dravet syndrome participating in this natural history study, if they take part in this study, then actually we have a standardized level of care that will be delivered across the United Kingdom, which I think, or which we think would be very good to achieve. And we know this from other diseases where this has happened when brought expanded natural history studies across a country where actually this leads actually to a better standard of care. So we're working jointly together with neurologists, child and adult neurologists, as well as neuropsychologists, and really and the collaborative effort across the United Kingdom. And what we were trying to do really to integrate at least part of the assessments into what you would normally have as a clinical assessment. So you would normally see your child neurologist maybe every 6 months. So in those assessments we standardize to make sure that that is the same across the United Kingdom. Next slide, please. So we were now in a position that we will hopefully start recruitment into the natural history study in the autumn of this year, and this will run over a period of 4 years. And the individuals will be followed up for at least a period of 3 years. Yeah, and any child or adult with Dravet syndrome or an SCN1A-related epilepsy can participate. Anybody with a new diagnosis or an established diagnosis can participate, and we have 300. We're planning for 300 participants of all age groups. Yeah, and these will be 240 children and 60 adults. And as Helen earlier explained, it's really important also for us to include adults because it's a disease across the lifespan. And it's really important to have adults. We don't understand really in terms of natural history study for adult Dravet syndrome individuals, and that's important. And this will be across the United Kingdom, so England, Scotland, Wales, and Northern Ireland. And if you're part of the natural history study, participants are also allowed to be enrolled in other studies and other trials. So this all goes hand in hand. So if somebody enrolls in the natural history study, that means they can still enroll in other medication trials or other trials that are taking place. The next slide, please. And so what that actually involves, taking part, means that you will have your clinical visits as part of your routine clinical care with your child neurologist every 6 months. And we will ask participants to 1 month before their appointment to keep a seizure diary of actually tracking seizure frequencies before the month that you attend. And then during the visit with your child neurologist, that will be a structured clinical form will be followed, will be filled in, really asking detailed questions about seizures and other aspects of health, about risks, about so that really is designed in a way it's uniform across the entire country. So really, and so asking about risk factors, for example, we're asking about SUDEP, about what measures are in place to actually address that. So it's really to cover the whole spectrum of what is really interesting and important to ask any patient with Dravet syndrome. And then we have the separate visits will be the neuropsychology visits, and these will be either 6 monthly or 12 monthly. So in the younger children, 6 years and younger, there will be 6 monthly. In the older individuals, 7 years and older, there will be once a year. And these are detailed face-to-face assessments with a neuropsychologist, really regarding learning and behavior, and as well questionnaires regarding comorbidities and quality of life. Because what we understand is that really often what is important for quality of life is not just seizures, but it's all the other aspects of life in terms of learning, behavioral difficulties, and all these other aspects. And we really would like to record this in great detail. Next slide, please. And so what we really hope to gain from this natural history study is that we have a better understanding of the epilepsy, but also of the what we call neurodevelopmental outcomes, so how the child or the adult evolves over time. And we would like to identify what we call biomarkers and predictors of the disease outcome. So that means, do we have any markers that can tell us how a particular individual will evolve and will develop over time? And that would be important to know. And by including individuals in the natural history study, we hope that we can raise the standards of care for individuals with Dravet syndrome and SCN1A-related epilepsies. And we would hope that being part of a natural history study network across the UK would then allow us seamless access to best evidence treatment approaches, because we're all going to be assessing children with Dravet syndrome and adults in the same way. Next slide, please. So that moves us onto the second part or third part of the talk, really talking about the disease-modifying and gene-based treatments in Dravet syndrome that have now become available. And as Helen said, we are really excited about this new possibility of treatment. Next slide, please. So just to lay a bit of the groundwork, why this is actually important and how genetic treatment actually might be helpful. So Dravet syndrome is caused by a mutation in the sodium channel alpha-1 subunit, so the SCN1A subunit. And the SCN1A subunit is a protein that is in the cell membrane of nerve cells, and that's really important for electrical signaling in the brain. And proteins, so it's a transmembrane proteins, and proteins are really the building blocks of the body. And the proteins consist of what we call amino acids, which are tiny protein parts that form then the protein. And we know that the SCN1A protein contains about 2009 amino acids, and the master plan of how to assemble each protein is contained in our DNA. Yeah, so our DNA really is a recipe for each protein in our body, so also the SCN1A protein. Next slide, please. So, and the DNA, here you can see this illustration on the left of the double strand of the DNA, which is really a code in each of our cells, and that code allows the body to form certain proteins. So it's like a recipe for proteins in the body. And here on the right hand side, you can see how that is all packed. So the DNA is packed into chromosomes and then actually located in the nucleus of our cells. Next slide, please. So, and you can really say that each DNA protein recipe is called a gene. Yeah, so what you can see on the left-hand side again, so you've got the DNA, and there's a section of the DNA that gives you the recipe to build a protein, and that's what's called a gene. Yeah, and so that gene is then what we call transcribed into what's called mRNA. You can see that on the top right, and that code is then translated into a protein at the bottom. Yeah, and this allows us, this is basically the principle. You have your DNA, which has your code and your recipe for your SCM1A protein, and that to be able to build a protein, the DNA has to be transcribed into the messenger RNA, and then the messenger RNA then helps to build via translation of that information into building a protein. Yeah, and what happens is if there's any mistake within your gene, then your protein can't be built. And what's important here is really that any gene-based treatment tries to address that. So they try to address that either at the gene level itself, at the DNA level, or they might address that at the level of the mRNA, the messenger RNA, for example. And this is some of the treatments that we're going to explain a little bit later on, really are based on those principles. And many of you might have, I mean, we've just been through COVID, and we had all these vaccines, and we had how the different vaccines work. And you've heard that, for example, the AstraZeneca vaccine works via the DNA, whereas the BioNTech works via the mRNA. So these are already terms that you would have come across in the media over the last year. So next slide, please. So what happens here is really, so there's different approaches for disease-modifying treatments. By disease-modifying treatments, we really mean treatments that will have an impact on which gene is expressed, how it is expressed, and how that might actually help to treat the underlying disorder. So what you can see on the top left is a printout of a genetic test, and that shows you, for example, that your child has a pathogenic SCM1A mutation, which is responsible for Dravet syndrome. So what happens here is that, for example, in your DNA, you have one piece of information that's mutated, so which means that the protein that's illustrated below can't be built. So, for example, you might have what's called a stop codon, is where I put the X. Suddenly, you can only build the first quarter of the protein, and because of the mutation, the rest that comes after the X can't be built. So you basically have a non-functional protein, and we always carry two. So we will have in terms of expression. So if you've got a mutation in SCM1A, that will reduce your expression of protein by 50%. Yeah. And that will then, so once you have a mutation and you only have 50% of the SCM1A when you actually need 100%, with only 50%, then what we see is that individuals develop seizures and all the other difficulties. And where we are now is that we can build models, how to model, for example, Dravet syndrome in Dravet mice or in other models. And we can then look at different treatments that, for example, RNA treatments or other genetic treatments and how they might work. So the next slide, please. So thank you. So this is really just an illustration, and don't worry if this seems very complex to you. So on the left-hand side, you have just this big circle that illustrates the different types of disease-modifying treatment that work in different ways. Yeah. So some of you might have heard about a particular approach that's called CRISPR. There's other approaches for example, where we've got gene supplementation, so where you give additional information that might actually correct a mistake that might be there, or I spoke about earlier about the RNA treatment, which is basically messenger RNA treatment, upon which you can also have strategies to modify the disease. And for example, in CRISPR gene editing, that's really based on the fact that the cell's genome, which means the cell's genetic material, can be cut at a desired location, allowing existing genes to be removed and new ones added. So it's a very smart technique to use that, but you can't use it in every cell. So there's another technique called CRISPR-A, which is a gene activation treatment. And that means that you make certain genes to produce more protein than they originally did. And this increases really the protein production in the body. And for example, if you then increase the SM1A production in the body, that might actually correct the underlying mistake. Next slide, please. So this is just to explain how this CRISPR gene regulation treatment actually works. So if you in the top right-hand graph, so you have what we call physiological. So that's in a normal person who does not have Dravet syndrome. So you have 2 copies of the SCM1A gene, and they're normally, if they're both produced, so that's the DNA, and that then produces the mRNA. And if you produce that normally, then you will have 100%, so 50% and 50% makes 100% of the gene of the protein is expressed. Yeah. So, however, in Dravet syndrome below, what you can see that one of those has got a mutation, that's a big X, and that's not producing any mRNA and any protein. So you're left with only 50% of the SCM1A protein. And if you have that, then you have Dravet syndrome. And what this CRISPR technique actually does, and that's right at the bottom, it actually enhances the expression of mRNA from one of the SCM1A copies, and they now produce double, and they produce 100%, meaning that actually, if you have a technique like that, that can actually rescue the defect from the SCM1A mutation. So this is really evidence from animal models at the moment for mice. And this hasn't been studied in humans yet, but this is clearly something that might be interesting in terms of treatment approaches. Next slide, please. So now coming to the treatment approach from the Stoke Therapeutics. So it's called targeted augmentation of nuclear gene output, also called TANGO. And that's, again, so targeted, so that's clear. Augmentation means there's an increase of the output of the gene output to rescue an SCM1A mutation. And that's based on the RNA, so on the messenger RNA-based treatment using something that's called an antisense oligonucleotide. And I'll explain that a little bit later. So this treatment is delivered via repeated intrathecal injections, and that means the repeated lumbar punctures. Yeah, so for this treatment to reach the brain, you have to give a lumbar puncture, and you give the medication via lumbar puncture. And this medication or this treatment aims to restore the protein levels by increasing the protein production from the one functional copy of the gene. So I explained earlier, in Dravet, normally you've got 2 copies. In Dravet syndrome, only one copy is working, and this treatment enhances the protein production from this one copy. And it selectively boosts the expression only in tissues where the protein is normally expressed. And this technique can be used for small or larger gene targets. And next slide, please, Helen. And this really just explains how this actually works. So you've got at the top, so on the left-hand side, you've got your DNA, and this is from your normal gene in green. We call that wild type, so that's normal. And on the right-hand side, you've got your red, you've got your mutated gene. And what we know from the mutated gene, that leads to loss of function. And at the end, you don't get any protein emerging from that. And what you see on the left in the green, so there's this process where you've got DNA, which is then transcribed into what's called pre-messenger RNA, so pre-mRNA. And then you've got a process that's called splicing. to produce actually productive mRNA, and that mRNA is then translated into the protein, and you see that at the right, on the right-hand side, on the left-hand side at the bottom. Yeah, so you can see that you have then the protein is produced. However, because 50% of your gene is not working, that's why you only get 50%, and you end up with Dravet syndrome. However, what's interesting here is that even in your normal gene copy, you have also after the splicing, there's one part of the mRNA which you can't use because it's got this blue bit in it that means that this piece of mRNA is actually going to be destroyed. And what you can see in the next slide is that so this treatment that Stoke developed called Tango, that actually stops the destruction of this part of the mRNA, so that on the left-hand side now you have actually double the production of your protein, which rescues the deficit that you had from the right-hand side. On the right-hand side, still, this is a truncate, or this is a copy that will not be produced and can't produce any protein, whereas on the left-hand side, now your protein levels go up from 50% to 100%. And what this means in practice, so this has been tested in mice. If you go to the next slide, please. So what you can see here, so this is a study where they tested this approach in mice. And the mice had a single dose of this. And what you can see on the graph on the left-hand side, there's the survival of mice over time. And what you can see here in green are wild-type mice. In yellow are mice that received this treatment. And in red are Dravet mice that didn't receive treatment. Yeah, so yellow. So what you can see is that there's a clear drop of the mice in red, which is the Dravet mice. So they had reduced survival, where actually the Dravet mice who received the yellow treatment, or here in yellow, they had increased survival. Yeah. So next slide, please. So where are we with this? With the current status of this particular treatment, so it's called the STK-001 trial. So the first in human dosing of in Dravet syndrome patients started with this medication last year in the United States, and it's currently a part of the Monarch trial. So that's what we call a phase 1/2 clinical trial, and that's currently ongoing in the United States, involving 48 children and adolescents between the ages of 2 and 18 years old with Dravet syndrome and a confirmed SCN1A variant. And this is now planned also to start in the United Kingdom, and it's called the Admiral trial. It's again a phase 1/2 trial planned in the United Kingdom now for about 22 children and adolescents ages 2 to 18 with Dravet syndrome and a confirmed SCN1A variant. And that means, so if it's a phase 1/2 trial, basically means that, first of all, the safety and tolerability of this particular medication has to be established. So we need to make sure we know that this worked well in mouse models, but we need to make sure this is actually safe for humans. So this is why we have phase 1/2 trials. And so the endpoints will be safety and tolerability of increasing doses, but also then measurements of seizure frequency and overall clinical status, and also quality of life. And so the aim is really to start recruiting later this year. Next slide, please. So the other treatment that is a GABAergic cell-specific SCN1A gene regulation approach. Again, so this is by a different company called Encoded, and this is a DNA-based treatment, and that treatment is delivered via a virus, an AAV9 delivered virus, and this is a one-time intracerebral ventricular, so that's a treatment that's given into fluid-filled spaces within the brain, and they're called ventricles, and that requires a surgical procedure to apply that treatment as a one-off treatment into the brain, into the fluid-filled spaces within the brain. And so this particular treatment has what's called a regulatory element that targets specific cells, GABAergic cells, which are inhibitory cells in the brain, and has an engineered transcription factor to increase production of SCN1A. And that leads, should lead to, or leads to upregulation of SCN1A expression within the target cell types of GABAergic inhibitory interneurons, and thereby correcting the mutation and the defect that we find in Dravet patients. Next slide, please. So this is just illustrating that. So this is on the right-hand side here, you can see the AAV9 vector. So this is basically the virus that has the regulatory component blue, and then the transcription factor in it, and that is then transported into the cell. You can see that on the left-hand side, and then that leads the cell in the DNA. What you have there, you have then an increased expression of SCN1A. And more increased SCN1A mRNA as a subsequently, and therefore more SCN1A protein. Yeah. So the effect is again here that you've got an increased expression of the SCN1A protein. And if you move to the next slide. Sorry, Helen. Thank you. So this is just a representation of this transcription factor, and that really leads to the increased expression of SCN1A protein eventually. Next slide, please. And what we can see is in the animal models here again. So this is, you can see how this medication prevents seizures and normalizes survival in the Dravet mouse models. On the left-hand side, what you can see is again here the percent of survival, and this is then over the period of time at the bottom. And you can see in the light blue, so in the dark blue are the Dravet mice without treatment. Sorry, I'm back. So in dark blue, it's the Dravet mice without treatment, and you can see that their survival is not good. There's reduced survival. However, in the treated mice at the top in light blue, you can see that the survival is much improved. And again, so this treatment also reverses the sensitivity to fever-related seizures or fever-provoked seizures. You can see here again that you have the percentage of seizures in dark here with the Dravet mice is increased, whereas actually the wild type and the treated mice are less susceptible to febrile seizures. Next slide, please. So where we now with this particular treatment, that's called the Endeavour trial. So the Endeavour trial again is a phase 1/2 trial, and that's planned internationally, including the United States, UK, Australia, and Spain, and about 24 children aged 6 months to 4 years with Dravet syndrome and a confirmed SCN1A variant. And again, because this is a phase 1/2 trial, so safety and tolerability of ascending doses, that's the main endpoint to make sure this is actually safe. And then also measuring a change in seizure frequency, developmental outcomes, behavior, motor function, sleep, and quality of life. And the aim with this treatment will be beginning of next year, in 2022. Next slide, please. That's so in conclusion, we've Helen very eloquently explained how we've got a number of increasing good evidence treatments available for Dravet syndrome, and now we have it's like the treatment algorithms are likely to change. So the treatments we choose for Dravet syndrome are likely to change over coming years with more evidence becoming available, and that there are some disease-modifying gene-based treatments becoming available. First of all, the Admiral trial starting this year in the United Kingdom, then the Endeavour trial starting next year in the United Kingdom, and then also the SCN1A Horizons UK the Natural History study starting later this year. And we really hope that with this study also we improve the standards of care of children with Dravet syndrome and SCN1A related epilepsy. And this is just the next slide. It's just a really to remember the Dravet Syndrome UK conference later this year to really to get further information on upcoming and new treatments as well. Thank you.
Well, thank you so much, Professor Cross and Dr. Brunkhaus, for that. That was incredibly informative. And something I would just say is I'm conscious there's a lot of information that's being shared on these slides, and probably for you all thinking, gosh, I need to go back and hear that again. So we have recorded this. Those slides will be made available on our YouTube channel within about a week, and we'll let you all know when those go live. So just say, don't worry if you've missed anything, there will be that chance to go over. So we've had a couple of questions come through into the chat. So if you haven't yet asked a question you'd like to, please do type it into the chat. So I'll just read out the first question. What if there are more than 22 people in the UK who are eligible for the Admiral trial?
That is a very, well, there probably are. I mean, it's a case that, you know, there are inclusion criteria, not least actually the number of seizures over a 2-month period. We know when we entered into the other studies, you know, that actually not everybody who thought they were eligible to enter into the study. So there's no doubt, and I've already, you know, you can see there are some patients that just won't fit the rise to the seizure criteria for that, let alone other criteria that may not make them eligible. But that said, there will be 6 sites across the UK who are recruiting. If, you know, we are making sure that professionals, or, you know, pediatric neurologists are aware of the studies. If people want to be referred in for consideration, then that's fine, but it has to happen through their local pediatric neurologist.
Thank you. The next question, fenfluramine has an EU license for 2 years and up. Are there any trials upcoming for 1 to 2 year olds?
There are not trials per se, but there's a lot of discussion about how that data will be acquired. And, you know, it's more likely, you know, the issue is getting data about safety. You know, once and if it is licensed, there's an issue about how that data may be acquired. But certainly it's something we're moving towards.
Thank you. Next question. Does limitation of Endeavour trial patients aged to 4 mean that this treatment is not expected to be effective in older patients?
No, it doesn't mean that. So I think they had to start somewhere, and so I think what we would there's no reason why this treatment wouldn't also be applicable for other age groups. So we haven't really seen any results yet in patients and what can be expected in terms of treatment effect, but we would imagine that, you know, if this is shown to be effective in younger patients, then there will also be a license to give that to older patients as well.
Thank you. Next question. Would other genetic mutations exclude participants from the genetic trials?
So this is, of course, it's a gene-based treatment. So these treatments only work if you have an SCN1A mutation. Yeah, so these treatments will not work for different for different genes. So these are specific SCN1A mutation treatments.
So next question: how long are gene therapy trials expected to last for?
Well, I think what we have to bear in mind is what I'm not sure is, the first thing I'll say is these are 2 studies coming online, and they're the first studies, and there will be subsequent studies, definitive studies later of both therapies, providing, of course, that the safety and tolerability is noted. So that's the first thing to say, and that's partly in relation to the 22 patients. You know, this, if you don't get into the first study, doesn't mean to say they won't be in the second study or the third or the fourth study. There are studies going to be rolling on after that. The second thing, how long does it last? That's a bit unknown. If we think about the Admiral study and the way that's done, I think it's actually 3 lumbar punctures over 6 months. And the difficulty is not knowing how long it will last after that. And it may be that intermittent lumbar puncture is required after that. Hopefully not frequently, but you know, that may well be required. We don't know yet. And it's presumed that probably, but not, you know, we don't know. I think with the encoded technique, at the moment, it's thought to be a single injection. But again, I think that's something that we have to find out over time. Correct, Andreas?
Yeah, that's correct.
Excellent. Thank you. Next one. Which treatment is more recommended, encoded or Stoke?
I don't think we can recommend one or the other. You know, we can just say these two trials are coming up. We don't know whether one is better than the other. They both have been tried and tested in the animal models. They both have shown increase of the protein or effect, but we have no way of saying whether one is better than the other.
Next one. Are the enrolment criteria for Admiral already known?
Well, yes. I mean, mainly based on seizure frequency. So you have at least 4 convulsive seizures over a 28-day period. They have to be on at least 1 anti-epileptic drug that's at stable dose for 4 weeks prior to screening. And all the other, it's like any entrance into any other drug study, really. Other drugs have to be stable for that. But, you know, those are the main criteria. And also, of course, between 2 and 18. And they've got a confirmed diagnosis of Dravet syndrome. And as per the previous studies, that is, you know, we report the clinical history and that's verified by a central place. And also, of course, they have to have a mutation in the SCM1A gene.
So the next question is, despite demonstrating clear Dravet symptoms, our neurologist won't confirm a Dravet diagnosis, and our son is diagnosed with SCM1A epilepsy syndrome. Would this exclude him from Dravet treatments and new therapies?
I think from the genetic studies, the issue is a clinical diagnosis of Dravet syndrome. That does not mean to say they're going to be excluded from other medical treatments, no. But it also doesn't mean to say that that in due course they be excluded from therapy. But with regards to these specific genetic trials, that is for electroclinical diagnosis of Dravet syndrome with SCM1A. If there's still a debate, they can quite happily ask for a second opinion, a referral in to see whether they're eligible, and then we can see whether they fulfill criteria for a Dravet syndrome. But as per criteria for the studies, they need an electroclinical diagnosis of Dravet syndrome.
Yeah, and I think it's important to know that, so there will be certain criteria to enter into the studies, certain things that have to be fulfilled, and it might well be that this child is actually fulfilling this, and it's, you know, for this you need to know the criteria and to see the child neurologist, the treating neurologist can then decide whether the child actually fulfills the criteria.
Thank you. Next question. Will there be a placebo arm on these trials, or will there be a delayed phase or crossover design so that all participants receive some form of trial treatment at some stage on ethical grounds?
Both of the studies to which we've reported are safety and tolerability trials, which means everybody who enters the study will get the treatment. Okay, there may be a dose finding phase, but everybody will get the treatment. The subsequent trials may well be placebo controlled, sham controlled. We don't know what that will be. But these initial studies are safety and tolerability studies, which means everybody gets the treatment.
And then we've had a follow-on point, the person who raised the question about if it wasn't an SCM1A mutation, and they said if it was in addition on top of SCM1A. So just asking for clarity if it's SCM1A but another genetic mutation in addition.
So I think what's clear is, so the main cause for the illness needs to be the SCM1A mutation. So that is clearly causing the Dravet, you know, somebody with an SCM1A mutation having Dravet syndrome, that would qualify for a genetic treatment. Yeah.
And so, Professor Cross, Dr. Brunklaus, we're coming up to 9 o'clock, and I've got some more questions here. How many more questions do you think you both can take before we finish?
I can probably do another 5-10 minutes. I don't know about you, Andreas. You're the one that's had a longer day than me.
Absolutely fine. Yeah.
That's very kind. And any questions we don't get to, we'll pose in email afterwards. So the next question is, the fact that we are discussing a dose escalating trial, does that mean that one should try not to be one of the first few who are being enrolled?
No, because the minimal dose that's shown to have benefit will be, and we're not giving a lower dose than wouldn't have been thought to be benefit or clinically beneficial, and there may be incremental doses through the treatment. So no, that doesn't mean that it doesn't. You don't need to be one of the first or the last to have any difference.
Next question: what results are being seen from the Monarch trial in the US? It's too early to say anything at the present time, other than to say there aren't huge safety concerns. That's all I can say. Okay. If phase 1-2 for Admiral goes well, will there be a phase 3 for a larger number of patients in the UK?
Yes, I mean, one thing to say is yes. We are the first country outside of the US to recruit. And if this all goes well, then we will be involved in the phase 3, yeah.
Is the trial scheduled for next year also a dose escalation trial? If not, is one better off waiting for next year than jumping onto the trial this year, but might be given an insufficient dose? I guess it speaks to the other question.
These are expected questions, of course, but I think again, so it's a phase 1-2 trial. So the Endeavour trial will also be looking at dose escalation, and this is about safety. So it's really, we have to establish in these trials that these products are actually safe to give.
Excellent. So I've got 3 more questions in the chat, and we're going to finish then on that third question. So the next one is, does age 2 to 8 include 18-year-olds?
No, once they reach their 18th birthday, they won't be eligible for these trials. They can be included if they're 17 and a half, for example, if they're under 18 prior to the time of recruitment. But if they are at their 18th birthday at the time of recruitment, no, they won't be eligible.
But I would say, I mean, if these trials are shown to be successful and working, then of course we would expect that this would then also be available to older patients.
Absolutely. Sorry, it's for these specific trials that it is 17 years, 364 days is the cutoff.
Okay, last 2 questions. Are there any planned Irish sites for recruiting patients?
Well, of course, Northern Ireland is included in the UK, and I presume they mean Éire, Southern Ireland. That is the site. I'm not aware that a site is planned, but we have been asked whether we would take patients from abroad, and at the present time we have said yes. And I had contact from Ireland today to ask that specific question, and I replied to that effect.
And it's the same. It's the same for us as well. So in Glasgow, we've had in previous trials also where we had participants from Ireland being treated in Glasgow.
Lovely. And the last question: can levels of SCN1A be actually measured in humans, or the effective treatment can only be measured via other observations?
So I think that's a very good question in terms of what you will measure here. So clearly you will measure, what is the effect of a treatment in terms of seizure frequencies, and then I'm not sure in terms of actual measurement of you should be able to measure SCN1A expression and how that's changed according with treatment. And what we find is, I think what we find is that the treatments, some of the treatments lead to 100% expression. There's an increase from 50% to 100%. For other treatments, they lead to an increase in expression, not unto 100%, but maybe to 90% or so. And even with that increase, you can see really significant changes in the mouse models, and that these mice actually have much less symptoms.
What I do know is from other genetic studies is that the actual amount is not correlated with clinical response. And so although you're saying you're increasing it, there's no target level because we don't know how much you need in order to see benefit. And in fact, you know, getting it really high, and I'm talking about other genetic studies, you know, can actually be toxic. So actually measuring it, although will happen to see if it correlates, there isn't a target that we know we can aim for. So you have to look for clinical response.
Fantastic. Well, thank you so much to both of you for your presentations today. That's been incredibly informative, and you know, lots of questions there that I think have really helped people's understanding. So we're coming to the end of our webinar now. Just a reminder, it will be live on our YouTube channel in about a week's time. So it's lovely to see everybody today on today's webinar. Thank you ever so much.